Hi, good morning. My name is Will Hwang, and I'm a physician scientist at Massachusetts General Hospital and Harvard Medical School. I'm going to share with you our work today looking at therapy-associated remodeling of the pancreatic tumor microenvironment using single-cell spatial transcriptomics. So before I start, I just wanted to acknowledge my entire lab, many of whom are pictured here. They did a lot of the work I'm about to show you in very close collaboration with the NanoString team, and I have their pictures later on in the presentation. So I'll start with some background. So I've been very interested in pancreatic cancer, both clinically and from a research standpoint, because despite pretty intensive research efforts over the last few decades, the outcomes are still very poor, with 5-year survival rates barely in the double digits, around 10 or 11%, depending on which statistics you look at. When you try to stratify patients and have an understanding of molecular guided therapy, you don't get very far with most patients looking at genomic data. There's a small subset of patients with actionable mutations. So the field, maybe about 5-6 years ago, started to turn towards cell state based on transcriptomic and proteomic information. And these three seminal studies that are pictured in the schematic here from Collison, Bailey, and Moffitt really split pancreatic tumors into two major categories. The first is known as the basal-like squamous or quasi-mesenchymal subtype, and the second is known as the classical or pancreatic progenitor. And all of this data was derived in bulk. So there was some question with some of these more minor subtypes like exocrine, ADEX, or the immunogenic subtypes, whether those were truly derived from the cancer cells themselves or if they were representative of stromal contamination. Nevertheless, these bulk-derived cell states had some utility. They seem to be prognostic. You can see here on the bottom left, this is progression-free survival in this Kaplan-Meier curve. And the purple classical line shows significantly better survival than the orange basal-like line. If you look at treatment, these are metastatic pancreatic cancer patients receiving multi-agent chemotherapy. And you can see from this waterfall plot that the classical patients in purple tend to respond better to multi-agent chemotherapy than the orange basal patients. There's still a lot that was not known when we started to look into this a few years ago. Specifically, I mentioned that the classical and basal-like cell states were found in bulk, but they were also derived from primary untreated resection specimens, a little bit of examination in metastatic samples, but largely primary. And as the field has shifted, there's more and more accumulating evidence, as you can see from this review, this table from a review, that shows how many various studies have demonstrated a potential benefit of neoadjuvant therapy, even in potentially resectable disease. So these are cancer patients that have pancreatic tumors deemed surgically resectable or borderline resectable, and there's a signal towards improved survival if you give them upfront chemotherapy, in some cases radiotherapy, or the addition of targeted therapy, or even immunotherapy has been looked at in selected subsets of patients. So given that most patients, by the time they enroll in clinical trials nowadays, have already been treated with chemotherapy at the very least, it's not clear whether these studies that looked only at untreated specimens are representative of the tumor states that are seen in those post-treated specimens that most of our patients bear. So to address this, several years ago, we started a study where at MGH, we have this large frozen biobank of primary resected pancreatic tumors. And a large number of them have been treated with chemotherapy or chemoradiotherapy, and some of them are treatment naive at the time of resection. So we pulled about 43 of these tumors, some of them untreated, some of them received CRT, which stands for chemotherapy and radiotherapy, and some received additional therapies, as shown in this schematic here. And we performed single nucleus RNA sequencing on these samples. This is the aggregate UMAP, showing that all the various major cell types that we'd expect in pancreatic tumors can be seen. And just examining one particular cell type, the malignant cells, we decided that it would be useful to do a de novo classification to try to refine the molecular taxonomy of these tumors. And we found that within the malignant cells, if you perform consensus non-negative matrix factorization and look for programs or cell states that are distributed across many patients, not just specific for one patient or another, we derived 7 robust cell states. And some of these recapitulated those bulk states that I mentioned on the prior slide. We saw a very strong classical program. The basal-like squamous or quasi-mesenchymal that were treated essentially as one cell state in bulk. With our improved resolution, we were able to separate those into 3 distinct programs: basaloid, squamoid, and mesenchymal. And then we found some additional programs. 2 of these programs, even though these were malignant cells based on various criteria, including having high inferred copy number variations, they had gene expression programs that were reminiscent of acinar cells or neuroendocrine cells, which are in normal physiologic pancreas, very important parts of pancreatic function. And then perhaps most interesting was this 7th cell state that we identified, the so-called neuro-like progenitor or NERP program. These are malignant cells that express neurodevelopmental programs, including certain genes that normally are only seen in the central nervous system. So to dig into this a little bit further, we looked, excuse me, we looked at our patients stratified by treatment status. So in this box plot, you're looking on the y-axis at the mean normalized program expression of the 7 malignant programs I just showed you on the prior slide. The white boxes are untreated, and then moving towards the more darkly shaded gray boxes, increasing amounts of treatment. And we saw reassuringly that the more treatment a tumor received, the less represented the classical program was. That is supported by prior literature. And on the opposite side of the spectrum, we saw that this newly identified neuro-like progenitor cell state, the more treatment you gave, the more enriched it became in the residual disease. And I'll just point out that while not significant in our data set, the mesenchymal program showed a slight trend towards being enriched in the post-treatment specimens. This has been looked at in other contexts. This is the closest among our programs to an epithelial mesenchymal transition, or EMT. And that has been shown in some contexts to be enriched in treated tumor. Now, this data, while we had about 43 patients, was not matched pre and post treatment. Those samples tend to be pretty hard to collect, even on clinical trials, because of the nature of pancreatic cancer and availability of biopsy tissue. So we decided to derive some matched organoids from a subset of our untreated patients. And here's an example. When you look at that patient's organoids and you treat with ex vivo chemotherapy and radiotherapy, you see there is a significant enrichment post-treatment of the neuro-like state. And then lastly, we went to 2 orthogonal data sets, TCGA and ICGC, and we looked at the bulk RNA sequencing data, and we looked at various clinical outcomes, such as time to progression, and we scored these tumors from the bulk RNA-seq for various malignant programs, fibroblast programs, clinical pathologic variables that you see here. And interestingly, we noticed that across these 266 patients, this multivariable Cox regression analysis showed that the classical program, again, not surprisingly, had the best prognosis, whereas our newly identified NERP program had the worst prognosis. So we felt like this newly identified program really had some clinical significance and was worth exploring more. So I won't dwell on this. This is from a paper that we published a few months ago. And this is a summary of the 7 malignant programs and the various features and associations and prognoses that we found correlated with each of these various cell states. So moving forward, this is some unpublished data that was led by an MD PhD student working with me and Andy Aguirre at the Dana-Farber, Jimmy Guo. So his thesis work really started to further dissect this neuro-like state. So we really wanted to understand this cancer intrinsic state. What was its clinical relevance beyond the primary disease? So he looked at some matched patient samples. This is before and after gem nab-paclitaxel. And in both of these patient tumors, you see a significant enrichment in the neuro-like progenitor state post-treatment in this matched setting. He also looked at metastatic disease and found that in metastatic disease, the neural-like state is even more enriched. It's actually quite rare to find cells that are predominantly neural-like in untreated primary tumor. If you go to metastases, you see a greater representation, and the most representing type of specimen is actually treated metastatic disease. So it seems like the worse the clinical disease, the more of this neural-like cancer cell state you see. Next, he started to look at regulators and biomarkers. There were a lot of, dependency map and other kind of computational analyses such as scenic, but ultimately he nominated and identified GLIS3 as a key biomarker and also transcription factor regulator of the neural-like state, and showed through functional genomics, such as the KP mouse model, he derived various organoid lines that use CRISPR activation to overexpress various transcription factors for different cell states, and showed that those largely recapitulated what we see in humans. And then took these in vivo with orthotopic transplants of GATA6, which is the master regulator of the classical program, the N-terminal deleted TRP63. TF is the master regulator for the basal-like, and GLIS3 I mentioned for the NERP-like was newly identified by Jimmy and our group. And when you orthotopically transplant pancreatic organoids into mice and look at tumor weight at 5 weeks, 8 weeks, 11 weeks, you can see that both the basal-like and the neural-like cancer cells grow faster than the control, which is has a scramble guide, as well as the classical GATA6 line. You also see more metastases with both lines. GLIS3, the neural-like line in particular, seems to have a predilection for going to the lung, which was interesting to see. And then lastly, we wanted to see what the treatment sensitivities were for these various cell states. And this is still ongoing work, but interestingly, the neural-like state seems to be more resistant to KRAS inhibition. And we're starting to look at various organoid lines, some of which are more neural-like than others, and looking at what their dependencies are to see if we can nominate other potential vulnerabilities that we can target. So that was really where we are now in terms of understanding these cancer cell intrinsic states and their relevance to treatment resistance. We felt like to take this to an even more relevant level, we really needed to put these cell states in the in situ context. So one of the first collaborations that we undertook with NanoString starting in late 2018, early 2019 was with the pre-commercial digital spatial profiling technology that's now the Geomics platform, of course. And we took some matched tumors. So of those 43 that we did single nucleus RNA sequencing, we performed digital spatial profiling at the whole transcriptome level for about 21 of those tumors. You can see how we serially sectioned them. We used the H&E slide to basically guide ROI selection. And here's a representative ROI. We use morphology markers against pan-cytokeratin for the epithelial compartment, alpha smooth muscle actin for the fibroblast compartment, and CD45 for the immune compartment. Segmented these. You can see the various areas of illumination, and we collected, whole transcriptome information from each of these cell types. And then we performed various analyses. So one of the more robust analyses that we were able to do was an unsupervised clustering, where we took the cancer cell compartment and we scored them for the 7 malignant cell states that I showed you before. We took the fibroblast compartment, we scored those for fibroblast programs that I did not have a chance to show you earlier. And then the immune compartment, we deconvolved for various immune cell type signatures that again were derived from the single nucleus RNA sequencing data set. And when we did this unsupervised clustering, the most robust clustering was 3 distinct neighborhoods, which we named based on the malignant subtypes that we saw. Treatment enriched featured the neural-like state with the EMT state, and interestingly CD8 T cells as well as these ICAFs, what we call the immunomodulatory CAFs in this study. Then the other 2 neighborhoods, one featured squamoid basaloid cancer cells, and the third one featured classical cancer cells. So we'll come back to this a little bit in a little bit. But one of the main limitations of digital spatial profiling, while it does have whole transcriptome coverage, it's limited in spatial resolution. So some of the things we wanted to do, you can certainly do cell-cell interaction analyses, but it's more limited when you're averaging across, 20 to potentially hundreds of cells, rather than looking at the single cell level. So we were fortunate to be able to collaborate with NanoString again through their early access program with their spatial molecular imaging technology, which is now commercially available as CosMx. And this really was what we needed to take another step. So this provides single cell, technically even subcellular resolution, spatial transcriptomics. It's not whole transcriptome coverage, but we've been doing close to 1,000 plex to 6,000 plex, which is quite good, as you'll see. And again, we took a subset of 13 tumors from the original 43. So for most of these 13 tumors, I should say, we have matched single nucleus RNA sequencing data, digital spatial profiling, as well as spatial molecular imaging. So here's an example H&E slide that we performed SMI on, and you can see how it lines up with the FOVs that were placed by the NanoString team pictured here. And I won't go through the technique because of Joe's earlier talk, as well as other talks where you've probably heard how SMI works. But if you look at how our samples performed, we were very pleasantly impressed by it. So on the bottom left here, labeled protein, this is the immunofluorescence image for one FOV. Green, again, are the cancer cells. The yellow is staining CD45, the red CD3 in the stroma. And then in the middle panel, we have made the protein stain a little bit more transparent, and we've overlaid as individual colored dots the localized RNA transcripts. And we've pseudo-colored those transcripts based on expected cell type specificity, green representing cancer, cyan representing genes we'd expect to be enriched in fibroblasts, and pink in immune cells. And you can see how nicely the architecture of the tumor at the protein level is recapitulated at the RNA level with the stromal malignant gland separation. If you zoom into this inset, the scale bar here is 25 microns. You can see that the segmentation of the cells perform quite well, and the based on the colors of the transcripts you see in each cell, you can see the specificity. At least at this level, is also appears to be quite good. So we of course did other QC metrics, and then satisfied with the quality of the data that was coming out through this collaboration. I mentioned we did this for 13 tumors, some with a 960 plex kind of base panel, which is very similar to the commercially available 950 plex panel, and then the 990 plex is where we added 30 additional targets that were designed to help us delineate fibroblast and malignant programs. And then we performed various neighborhood analyses and cell-cell interaction analyses. So you can see across our 13 samples, we had over 700,000 cells in situ that passed quality control metrics. When you look at this UMAP and gene expression space, the gray non-malignant cells and the green malignant cells separate pretty nicely. And this was also a really pleasant surprise. When you look at this dot plot, you can see that the various marker genes along the horizontal axis here really pretty cleanly separate the various cell types we expect to see in these pancreatic tumors. On the bottom here, bottom left, with 990 genes, we were not able to really delineate all 7 malignant programs with high fidelity, but we were able to separate the 3 of greatest interest to us. So while it's a simplification, we took the bulk defined classical and basal-like programs, and then our newly identified neuro-like state, and we were able to separate those quite well amongst the malignant cells. And then similarly, the well-established myofibroblastic CAFs or MyCAFs and inflammatory CAFs or ICAFs that were first defined by David Tuveson's group, those separate very nicely as well. So we moved forward with these annotations, and one of the first things we were interested in doing was to look at some of the cell proximity data that we had from geomics, but that wasn't single cell resolution. So we were curious which of those findings would hold up. So you can do this. You can do this for any kind of pairwise combination of cells, which you can see in this heat map here. And we did this by leveraging an exponential model. So I'll show you an example here. We were really curious about that CD8 T cell association with the treatment-enriched neighborhood that I showed you earlier, because we know that having more cancer cells that are post-EMT and apparently that look neuro-like in their expression profiles, that portends a poor prognosis. But we've also seen in many other cancers, including pancreatic cancer, that having higher tumor-infiltrating lymphocytes seems to be a good prognostic factor. So we were surprised to see the CD8 association with these two kind of bad cancer cell types. So we wanted to check this at the single cell level, right? So what we did is here you're looking at one FOV, and we mapped out where all the CD8 T cells are, and we modeled each CD8 T cell as an exponential function, where the decay radius, you can vary that, and the results I'm about to show you are robust to a very wide range of decay radii. And this underlying topology shown in red is based on the summed exponential functions from the CD8 T cells, right? So the higher peaks represent a higher density of CD8 T cells. Then on top of that, we've mapped as individual colored dots malignant cells that have been annotated as classical, basal, or neuro-like. And when you compare the location of these, if you look at each of these cancer cells, and you essentially sum those underlying exponential functions from the CD8 T cells and compare them to a null distribution where we keep the proportions of malignant annotations the same, but we scramble their location, we found that the log-fold change of the observed distribution over the null distribution was significantly higher for the neuro-like state, significantly lower for the classical-like state, suggesting that neuro-like cancer cells are indeed located more proximal to CD8 T cells than you would expect by random chance, and classical cancer cells are located further away from CD8 T cells than you would expect by random chance. As another example, we looked at a finding that was not from our own group. This is from Dave Tuveson's group, published in 2017, when they identified these ACTA2 high MICAPs and these IL-6 high ICAPs. They found that the MICAPs were located in closer proximity to cancer cells than ICAPs. So we wanted to see if that was borne out in our data. So we did the same method with this exponential model, and we found that sure enough, MICAPs were significantly more proximal to cancer cells than predicted, and ICAPs were significantly further away from cancer cells than predicted. So take this another step, right? So we looked at cancer intrinsic cell states that are associated with therapeutic resistance. Then we looked at the in situ context. What cells are in the same neighborhood as these various cancer cell states? Now we wanted to take a look at what interactions are maybe occurring between cancer cells of different states and the stroma that surrounds them, and how that might actually lead to emergent properties such as therapeutic resistance. So in close collaboration with Martin Hemberg and his postdoc Jingyi Cao over at Brigham and Women's Hospital, we developed this method called spatially constrained optimal transport interaction analysis, or SCOTA. You can kind of see the schematic here. So this builds off of some prior methods that had used optimal transport, but with a few differences. So we start out by identifying adjacent source and target cluster pairs. So any two cell types, you can run this analysis. And we hypothesized that even with paracrine interactions, clusters of source and target cells that are closer together are more likely to communicate than those that are very far apart. So we use dbScan to help us nominate these kind of adjacent source and target clusters. And then from there, we designed a cost matrix that factors in spatial coordinates of the source and target cells, as well as expression of the source ligand and the target receptor. And then perform optimal transport analysis on this, and you end up with this matrix of spatially constrained cell-cell interaction scores that you can perform downstream analyses on. So when we applied this to our treated samples and compared it to our untreated samples, you can see in this core diagram that the most enriched interaction was between calf source cells and malignant target cells. You can see how much thicker the cord is in the treated diagram compared to the untreated. And when we looked at this in greater detail, in this volcano plot, you see these red dots here represent significantly enriched predicted interactions in treated tumors, and the purple represent those that are significantly depleted, okay? And I'll come back to some of these specific examples later, but just to kind of do a visual sanity check, here's one particular interaction that's predicted by Scotia to be enriched in the post-treated specimens. CLCF1 is the ligand, CNTFR is the receptor. This is in the IL-6 family. Here are 4 representative FOVs from 4 different untreated samples, 4 different treated samples on the bottom, and you can see visually that there really does seem to be an enrichment in this interaction in treated specimens. Now, we didn't want to just look at this in one relatively small data set. We wanted to look to see if there was a correlation with our SNOOC-seq data, for example. So on the x-axis here, this is the Scotia results, the interaction fold change between treated and untreated. On the y-axis, this is the ligand, we did ligand receptor analysis on the single nucleus, non-spatial data. This is the exponential fold change between treated and untreated. And you see there's a significant positive correlation. We also posited that if a predicted interaction is really happening, right, we just have a snapshot of the tissue frozen in one point in time, we would expect to see evidence downstream of the receptor of activated targets being positively correlated, repressed targets being negatively correlated. So when we did this across all of the interactions, and leverage the KEGG and TRUST version 2 databases for the downstream targets, we saw indeed there was a significant positive correlation between these interactions and the receptor activated targets, a negative correlation with the receptor repressed targets. So that was yet another check that this method seems to be pointing to real biology. As yet another complementary method, we derived ductal organoids from the KP mouse model. We co-cultured them with pancreatic stellate cells, which differentiate into cancer-associated fibroblasts when cultured with cancer cells. And then we treated some of these with ex vivo 5-FU, which is the backbone of the FOLFIRINOX chemotherapy that our patients get. And we did single nucleus RNA sequencing of the treated versus the untreated specimens, which you see here in expression space in this UMAP. When we compared again on the x-axis the SMI interactions with now the co-culture interactions, we again saw a significant positive correlation. And one of the hits that really pops out as one of the top hits over and over again is some of these IL-6 family interactions like CLCF1, CNTFR. This is just another way to show on the left side of this panel, these are the enriched hits. You see pretty good concordance between SMI and the co-culture, the murine co-culture. And on the right, you see the depleted interactions, again, with a few exceptions, pretty good concordance across these 2 model systems. So just a couple, in the last couple of minutes here, a couple tidbits of biology that we found very interesting. So this is just me showing that initial volcano plot again of the enriched and depleted hits in association with treatment. We found that there were a bunch of collagen integrin interactions that were depleted post-treatment, but also these Wnt interactions where the calf is supplying the Wnt ligand, and that signals through various Wnt receptors such as Frizzled-5. And post-treatment, we see that there seems to be much less dependence on this, suggesting that maybe cancer cells that survive chemotherapy and radiotherapy become a largely kind of niche independent from the calves in terms of needing them to supply Wnt ligand. Further supporting this, if you just look at the mean expression of various Wnt ligands in the fibroblast post-treatment, there's a significant decline. And then in the post-treated cancer cells, there's a significant increase in expression of Wnt ligand. So it supporting this hypothesis. I also mentioned IL-6 family signaling. This story is still being worked out, but our group and others have seen that after chemotherapy and radiotherapy, there's an enrichment of ICAPs at the expense of MICAPs. We find that actually neural-like cancer cells, and to a lesser extent basal-like cells, are more proximal to ICAPs than classical cells. Going back to the DSP data where we had whole transcriptome information, so we were able to parse out all 7 of our programs, we see, interestingly, there's only a significant positive association between ICAP proportion in the DSP ROIs with mesenchymal cancer cells and neural-like cancer cells. The neuroendocrine is weakly associated. That's related to the neural-like state. And not to belabor the point, but we think that the type of IL-6 signaling, whether it's classical or trans, may actually dictate in part what a cancer cell state becomes, suggesting that there may be some plasticity in cancer cell state driven by signaling through the IL-6 family from neighboring ICAPs, especially when we're talking about basal-like mesenchymal cancer cells and neural-like cancer cells. And this is very much a work in progress that we are hopefully going to be able to report in more detail in the coming months. All right, so for the sake of time, I'm going to jump over this. This was an orthogonal point I was going to make about these programs and another study we're doing looking at cancer nerve interactions. And I'll just end by saying that right now we have among some other exciting collaborations, we're lucky enough to have some early access to 6,000 plex SMI, which really is designed to look at neuroscience, immunology, and cancer. And this is really getting at some of the very interesting cancer nerve stromal interactions and immune interactions that we're starting to detect may play a very important role in therapeutic resistance, metastasis, and other things. And this data so far looks quite good. Out of the 6,000 genes, we're detecting about 5,000 above the limit of detection. So we're really excited that the analysis is ongoing right now. And in terms of future stuff, beyond kind of the more obvious integration of high-plex protein and RNA, we're very interested in spatial analyses of matched pre- and post-treatment specimens. We have clinical trials now that are going to feed directly into some of these technologies and methods that have been developed in collaborations. And then we're also interested in doing more novel things, such as spatial genetic perturbation screens, looking at cell-cell interaction analyses beyond ligand receptor, doing pseudo-time trajectory analyses that account for spatial relationships, and perhaps actually starting to improve the Z resolution so that you can do spatial analyses on thick specimens without serial sectioning. And with that, I'd like to thank all of the members of my team for their amazing efforts on this work and other work that I didn't get a chance to show today. All of our collaborators, my clinical collaborators, my former postdoc mentors, all of our funding agencies, and in particular, the amazing NanoString team for being wonderful collaborators over the last 5 years. Thank you very much.